Our next speaker is Sandra Schaer. I'm talking about from Biognosys AG, talking about the digital proteome.
Thanks a lot. Welcome to the Digital Proteome. Pharmacological drug development usually starts with identifying a biological target and then advances through a multidisciplinary process in order to identify a safe and efficient therapy. Insights from human disease and biology help designing preclinical model assays where drugs are being tested for its safety and efficacy. And these are then further leading into the design of human clinical trials. But nowadays there are still quite some major knowledge gaps in translatability between human and preclinical species and then further back to human. By using in silico and ex vivo models, one already tries to improve translatability. And by using genomics and transcriptomics databases, people are trying to do the same. But at the end, in most of the cases, proteins are the biological targets, as proteins control the biological processes. So now let's have a look at this GPCR receptor in humans. We would now like to do and test some drugs in preclinical species. And we are potentially getting outcome A, B, and C. And then we are translating it back to human and we are getting completely different outcomes A, B, and C. And why that? Reasons could be that the sequence, so the amino acid sequence of the GPCR receptor from human to the preclinical model is different, or we have different post-translational modifications. Or at the end, the protein of interest is not only expressed in the tissue of interest, but also in other ones, and therefore leading to unwanted effects in human. So how can we improve translatability? That's by doing a deep proteomic data set. And that's exactly what we've done. We selected 5 species, human, mouse, rat, dog, and mini pig, and selected about 25 tissues per species. We then did a bottom-up proteomic sample preparation and went into 2 directions. One direction was doing deep fractionation and the other one was doing single-shot sample measurements. With the deep fractionated samples, we did DDA acquisitions in order to get a qualitative data set. Whereas on the other hand, with the single-shot samples, they were measured with a DIA method in order to generate a quantitative data set. But now let's start with the qualitative data set first. Here we selected a deep fractionation method, in our case high pH reverse phase chromatography. Each tissue sample was fractionated into 60 fractions, whereas only every second one was measured on the mass spec. At the presentation 2 years ago at ASMS, we showed that by doing deep fractionation, so in total 60 fractions, and only selecting every second one, you can see deeper into the proteome instead of just doing 30 fractions. We then selected the DDA method where we used FAMES and combined 4 compensation voltages. Spectronaut was used to do library generation. And now I would like to give you some numbers. We have 5 species times 25 tissues. That leads to 125 tissue samples. After fractionation, we ended up with almost 4,000 samples. And by using a 2.3-hour LC-MS method, one mass spec was basically running for more than a year. And we ended up with roughly 5 terabytes of spectral data. So now I'm happy to present you the digital proteome. Let me guide you through. So we start with the human samples. This is at the outermost part, you see a visualization of the identified protein groups per tissue, whereas we have the most protein groups identified in testis tissue and the least in muscle. To give you an impression what depth we actually achieved, we were looking at the human pathways. We gathered those pathways into roughly 10 buckets and checked how many proteins we actually identified. and we identified more than 90%. So the proteins we actually identified are just not any, we actually identified pharmacologically relevant proteins. Let's go back to the main figure. We were able to identify between almost 18 and more than 21,000 protein groups, and in average about 850,000 modified peptides. Also here we were asking the question, how can we then actually use this data set to improve translatability? And for that we mapped the orthogonal proteins from mouse, human, rat and dog to the ones of human. And theoretically one could identify more than 14,000 proteins. In our data set, we reached more than 80%. So also here, that means we can actually use this data set to improve translatability. So you can check the data set for certain proteins and see where they are expressed in what tissues and across what species. Let's go back to our example here. And now I would like to go back in time to the year 2006. Theralizumab case, you've probably already heard of that. This antibody was tested for its safety and efficacy in monkeys. Everything was fine, therefore the drug was administered to 6 volunteers, and all of them needed immediate care and some even had multi-organ failure. So what happened? Later on they figured out that the CD28 receptor on CD4 cells is actually downregulated in monkeys, and therefore the immune system in humans was acting completely different. So what does this tell us? It means that it's not only important to know where the protein is expressed, it's also important to know at what level. So therefore we generated a quantitative data set. Again, using our 5 species times 25 tissues. We did bottom-up proteomic sample preparation and measured those samples directly with a DIA method using timsTOF HT. Also here to give you some numbers, we have 5 species times 25 tissues. Important note, for all preclinical species we had matching tissues from male and female. This resulted in a total of 200 tissue samples that were measured with a 40 samples per day method. That means within just 5 days we acquired all samples. We were able to identify between 11 and 14,000 protein groups and between 160 and 220,000 modified peptides. To quickly compare those 2 data sets, we are now looking at the human tissues, and let's start with the depth. Here, with the DIA data set, we reached more than 50% of the human proteome coverage, and with the DDA data set, we reached almost 90%. Therefore, this is also one of the deepest proteomic data sets ever created. Then in terms of speed, the DDA dataset took quite some time, in total more than 80 days, and with the quantitative dataset we just did it within 1 day, so super fast. So let's again have a look at the human tissues, and I would like to give you some results on the protein level. Here we selected some kinases. It's well known that kinases are involved in cancer development. We see them listed on the y-axis, and on the x-axis we have the human tissues. With this heat map we see that we can actually identify and quantify these proteins across human tissues. But let me go one step further. We will now have a look at the peptide level. Here I've selected one specific family of kinases, the casein kinases, again on the y-axis, and here it's actually apparent that we have multiple isoforms. And it's quite cool to see that we were also able to identify and quantify different isoforms across human tissues. And I would like to highlight casein kinase 1, delta and epsilon. These have a high sequence homology, about 97%, and we were still able to show or see the difference. Now let's continue and go again back to the database. We saw that we have protein and peptide level information, but now let's compare across these species. Now, comparing protein quantities across species is not that easy, and we wanted to avoid having an additional sample preparation step. For example, the need of adding heavy labeled peptides. We were looking for an easy way. And here we selected the proteome ruler. The proteome ruler was published 2014 by Wisniewski. And what it does, it takes the relative quantification data of proteins and normalizes it to the histones. So histones exist in a tightly conserved ratio among mammalian systems and have a clear ratio to the DNA. So by calculating the concentration and therefore comparing it to the protein size, you can actually calculate the relative protein copy numbers per cell. So let's have a look at the PCA plot. Here, the different tissues are colored differently. So here we see that brain and heart are the most distinct tissues, which is already quite well known. But now let's go one step further. Here we are looking at the tissue samples which are colored based on their species, and we can actually see that they are overlaying, so we don't have a huge difference among these, which is actually what we want. And now let's come to the, in my opinion, the most exciting data, the PCA plot, where we have the different tissues colored. So per tissue we have a color, and we see that we actually have tissue samples that are clustering together independent of their species. And to show you this amazing data in a slightly different plotting strategy, we are now looking at the UMAP. And here, I'm super happy about this data because we have heart, we have brain, liver, testis, bladder, thyroid, and more tissues that are clustering together independent of their species. So that means those all have similar relative protein copy numbers per cell. And I would again like to stress that this data is just coming from one sample per tissue per species and already looks so great. So imagine just adding more samples to the data set, you could increase the statistical power massively. And with that, I'm already at the end of my presentation and would like to conclude what we just learned. Let's start with the DDA data set. Here we see we can now use this for biomarker discovery. Further, it can be used to increase and support translatability across species. On the DIA dataset, this actually gives us protein and peptide level information. Further, it shows us correlation, so you can do evaluation, you can get correlation among tissues and species, which is super cool to do. And now let's have a quick outlook to the future. So this huge and deep peptide database can now support SRM or PRM assays. So you can actually select certain peptides which are useful for targeted assays. And on the other hand, you can also use this data set to do explorative analysis. So you can search for the dark proteome. You can have a look what's behind the moon. So the possibilities are huge, and I think this data set is a great baseline to start and improve the translatability, and I hope we can actually support pharmacological drug development with this data set. And I would like to thank everyone that was involved in this project. This was a huge team effort, and thanks a lot for listening.
So, questions? That was epic. So.
That's a great talk and a valuable data set. I saw you mentioned that between the during the cross-species comparison, the caption says you only compared like the protein are missing value free across all species.
Yes.
So I was wondering how many proteins was involved in this cross-species?
It was about 2,000 proteins involved. So we filtered for only proteins that do not have any missing values, just because we wanted to avoid having doing an imputation strategy. But you can, of course, do certain imputation strategies and would probably get to the same result.
So does that mean that only like about 10% of the proteome depth you quantificate are involved in the cross-species?
Yes, for this analysis, we were really highly filtering the data set.
Okay, thank you. Thanks for the explanation.
Thank you so much for sharing this important tool. I think this is a game changer for drug discovery research. Given the discrepancy that you described between human and non-human primate for one of the proteins, but I didn't see non-human primate listed as one of your target organisms, is there a plan to incorporate those tissues as well?
Yeah, thanks a lot for this question. We are actually planning to incorporate this as a next species still this year.
Okay,
so
great,
it's coming. So keep heads up.
Great talk. That was really cool. For your single shot quantitative for the human, so you have about 150,000 modified peptides.
Yeah.
What are the modifications that you looked at? And also, if you looked at only strip sequence, what would that number be?
So the modifications were acetylation, carbamidamethylation, I believe,
Not phosphorylation or?
No, not phosphorylation.
And what would the number be if you only look at the strip sequence? So get rid of all the modified.
Peptide-wise, I'm not sure actually.
Okay, thank you.
Great talk. How do you handle the FASTA databases for some of these preclinical species like dog and pig? We find that there are a lot of isoform X1s, isoform X2s, and so the protein grouping algorithm kind of struggles with collapsing everything down to a single group.
Not sure whether I understood you correctly, but so it's quite difficult to basically match different proteins and compare these among species, just because one big issue is the FASTA file annotation. So for example, human and mouse FASTA files are pretty well annotated, but for like rat, dog and mini pig, it's rather difficult. Therefore, this is already a first challenge to tackle this. But I think working on that would already help a lot to improve the overall coverage.
So do you guys have a way to handle those pig databases that have a lot of redundancy and isoforms?
This is also something we will work on further this year.
Great. Thank you.
So we have about a minute to stay on schedule. So I'm curious, male, female, you have all that data. Have you started down that path? Yes, we can see differences actually, so it's a little bit sad we don't have male and female matching tissues in human, and I think that would be a great addition. So also there we would like to add these, but for male and female you can actually see differences for certain proteins, so that's super interesting. And I guess I'll keep going. Organs that are the most different. Sorry? So the organs that were the most different across the species. The most different. So I mean, based on the UMAP, I think, here again I need to say that just based on this one sample per tissue per species, it's quite difficult because the sampling could have been completely. So the most different was actually the eye, just because there for one species the full eye was processed, and for the other species just the retina part was processed. Therefore, we could see these huge differences. Super. Thank you so much.